Left off talking about building up DNA, building up DNA and RNA from basically the very basic structures, kind of like for enzymes, we first looked at how amino acids are connected together. And we had built up to the point where we had nucleotides, which was a base plus a sugar molecule plus a phosphate. And so the next step is to combine that series of molecules together. And so what we're really going to look at, oh yeah, I put these in here so that if those of you that didn't bring your book, we'd actually have some pictures to look at. Let's see, we got to scroll all the way past them. Still there, still there, still there. Okay, looks like that's fresh slides starting on number 13. And so what we're going to see is that if we kind of think about it, depending upon how you draw it, we usually go up and down just because it's easier to draw the molecules that way. But you're going to kind of have something that looks like this. And I'm going to sketch it out kind of like that. So P is standing, of course, for phosphate. And D is going to be our deoxyribose. And so the backbone is kind of made up of alternating phosphate deoxyribose molecules. Oh, the bottom one? Yeah, I was in the middle of writing, so D, yeah. So the deoxyriboses, and then off of the deoxyriboses is where we have our bases. So that's where you're going to have your A, G, C, T parts. And so it's kind of continuing on that. And if we add a little bit more detail to it, what we see is that it goes, let's see, if the way I've got it drawn, this would be the 5' end, and this is the 3. And so the phosphates are connected between the 3's and the 5's. And so this phosphate would be on the 5 of one and on the 3 of the other, on the 5 of one and the 3 of the other. And so we can liken that to sort of how we had chains of glucose molecules for carbohydrates. Lipids don't really follow. They don't make big, super long chains. That's kind of like maybe more phospholipids and things like that we can look at. But it's very like what we looked at with amino acids, you know, where we'd have big, long strings of amino acids. We could write it like that, or we could realize that what we really had was always the carboxylic acid end, a CH, an NH, and then that was to a carbonyl group, to a CH, to an NH, to a carbonyl group, to a CH, meaning that that pattern repeats itself. And then hanging off of here is where we had our different R groups that told us what amino acids we were connecting together. And so part of the molecule is a very repetitive nature, meaning essentially those amide bonds that are holding the proteins together. And we can liken that to the alternating phosphate deoxyribose in the DNA molecule. So if we take a look at an example of how we would do that then, so here's our ribose. And then on the 5' one, you would have your CH2 to an oxygen, and then that's going off to your next phosphate. And then off of here is, of course, where we hang our base. And I'm, again, not going to bother drawing the base. You can practice that on the homework, and you know, I don't think you're learning anything by drawing it over and over and over again. And so then that is on the 5' one again, so I guess if I'm being consistent, we'll draw it like this. This is a pain in the butt to draw, is all I gotta say. Dash O, dash P, double bond O, dash O, minus, minus, make that look nicer, double bond O, dash CH2, and so on. So here's another base, here's another base. So I want you to kind of get that idea of alternating parts. So this is an example for DNA. How do I know I drew DNA? Yeah, no OH. If I wanted to draw RNA, I would just put in OH there, right? So that's how the strand of DNA is made. It's simply that repeating set of units over and over and over again ad nauseum. And of course, there's about 50 million bases in the human genome. So you can imagine, you could draw this for a long time, maybe the end of time. You guys are also probably well aware that DNA is complementary. What does that mean? Yeah, it's really composed of two strands that are opposites of each other. Or really, we should say complementary to each other. So the two strands are complementary, or composed of two strands there, and forms a double helix. That should be basic high school, college biology. And so what do we mean by that term complementary? Well, the way they're complementary is they're held together by hydrogen bonds. So that's held together by hydrogen bonds between base pairs. And so what I'm really saying is that if I take a look at this picture, that base would be hydrogen bonded to another base. This base would be hydrogen bonded to another base on the other strand of the DNA. And here's where Jay's drawing skills stop, and here's where your drawing skills get to stop too. Meaning, as long as you can put together one half of a DNA strand, putting together the other half and showing the hydrogen bonding between the base pairs would be honestly just a pain in the ass. Ooh, I said that on tape. So instead, let's just take a look at it in a better looking picture from our book. So you can see that, for instance, here is a strand, and it's similar to what I was trying to draw. DNA or RNA? Oops, RNA, RNA. So remember, that's the RNA. If that's missing, like in our picture that we drew before, that's the DNA. What else do you notice is different about this picture? What? Oh, so yeah, these are of course our bases, and I was lazy and didn't draw those, right? So this is a better picture, obviously. What other little difference do you notice? Something we've talked about before. I even started circling it. Yeah, so sometimes, remember, some books will show phosphates like this, and in some parts of the books they like to show phosphates like this, where they have an O- for one of them. Or, and the other two are connected to other things, obviously. So notice that in the lipid chapter, they showed that O not having a hydrogen attached. For some reason, in this slide that I have, they show that OH that H being attached. And what I find hilarious is, since I have this open to the exact same page in my book, just by chance, that in this version of the book, they have it as O-. So even in different versions of the book, they're going to draw it differently. So that's just one of those things I want you to note, so that if you look somewhere else on the internet, you look in your book, whatever, and it's a little bit different, that you're not like going, oh, well, that's totally different and wrong. Well, it depends obviously on how the book likes to go about doing it. The other thing that you could notice, not in so much, well, actually, there's one other thing that you could notice that might tell you that this is RNA versus DNA. And I have to be honest, I haven't covered this yet in lecture, but most people from biology know it. What would be the other big difference between DNA and RNA? true. RNA is single, DNA is double. Yeah, the U here means that it's RNA. So remember, RNA is AGCU, DNA is AGCT, and I made my C and my G kind of look the same. I don't know why. Probably because I'm not very good at writing. So what we mean by that complement range, sorry, I kind of got off topic when I saw that picture, is sort of this idea of, showing it forms a double helix. But what it's showing here is that that double helix is held together by 3 hydrogen bonds between a G and a C, and then A and T's form 2 hydrogen bonds between them. And so by the way that that DP strand works, it naturally has a twist in it. Just kind of like, for instance, when we talked about the glucose molecules being stuck together, if they're just alpha-1,4 glycosidic bonds, that formed a normal spiral too, right? Just like for amino acids, we said that if you have the right set of hydrogen bonds between the secondary structures, that forms that alpha helix, right? So this helix structure sort of pops out naturally, although for different reasons in each of the cases. Glucose, it's different bond angles, different bonds and everything like that. The amino acids are different, and then this is different. To me, it's one of those just amazing things. It's like we can get helixes in 3 different ways. So it must be one of those, I won't say it's a universal shape or anything, but it certainly must be one of those highly idealized shapes that, if something can naturally form a helix, it's advantageous. So in this case, those D's and those P's start to make that helix, and then the 2 helixes are connected together by the hydrogen bonds between the base pairs. Think about this also. This is kind of like how we were showing it with enzymes and proteins, in that, instead of I guess an analogy to the proteins would be that it's the interaction between the R groups that led to, other types of shapes. Although for proteins, it's actually the secondary structure that causes the helix. Here, I guess maybe that isn't a very good analogy, because in one case it's the R groups, and in one case it's the base pairs, and they're not exactly the same. But I think it's beautiful. I think it's amazing that somehow all these molecules line up just perfectly for this to occur. Whether that's random chance or some higher power, well, that's up to you guys to decide. So here's another example showing it. This is a kind of an idealized structure. They're not actually showing the bonds or anything like that. This is supposed to be our deoxyribose. This is a picture or a symbol showing the phosphates, but it is again showing that complementary nature between C's and G's and T's and A's. And then this is an actual picture. Like, see, for instance, could you imagine me trying to sketch this by hand and getting all those things to line up just perfectly? I guess if I was really into this and this was my favorite topic in the entire world, I'd master drawing it. But I'm not going to learn to draw it. I don't expect you guys to learn to draw it. I expect you to mostly remember that you can form 2 hydrogen bonds between an A and a T, and you can form 3 hydrogen bonds between a C and a G. And it just so happens that those shapes are perfect for forming them. That's the other thing that gets me, is just how perfect they kind of have to be in order to do that. Meaning, change a single bond and those hydrogen bonds aren't going to line up. Make a single bond a little bit longer or a little bit shorter, and they're not going to form that perfect helix structure. So just all of the little things that had to come together to get that to work out like that, to me, is fantastic. I just can't believe it actually functions. Can you sing it?
I don't want to.
Well, you shouldn't have brought it up.
Well, I was just weird that it just sticks in your head. It actually helped, but it was just to like real.
So what's it kind of sound like? You don't have to sing. You can just say the words.
I'm gonna sing it. We love you, beautiful heights, phosphate sugar and the base, falling down to sides. Adenine and thymine make a little bit. Guanine outside seed with the
I love it. That is awesome. But see, do you remember this shit?
Yeah, that's how I learned about these things.
Yeah, I was talking to Jess while she was studying, and she brought up, I don't even remember what it was. She was like, oh, is this like something in Bio 111? I'm like, shit, you remember something from last
Oh, yeah, that's
semester?
from my freshman year.
Freaking amazing.
That's from my freshman year.
Yeah, and a certain part of me, I remember reading a study saying that anytime you can put something to a song, a picture in your head or something like that, those things stick longer.
Yeah, you made us sing in a bunch of class.
Well, this is college. We don't get to have any fun like that singing. Plus, if you think she sings bad, you should hear me. So, anyway, I want you to draw attention to that complementariness of it. Realize that that's how the DNA helix is formed, by the hydrogen bonds holding it together, but don't draw a picture. That would be excessively bad or tough or ridiculous. It's a little bit even more complicated than that, and maybe I don't have a picture in it because I took it out apparently or never put it in. We'll just assume it's not in there. It might be somewhere else. I can refer to it in your book, though. That's pretty easy. There's a lovely picture that's kind of on page 862, figure 31.9. It kind of goes into the larger scale structure, which talks about how the DNA strand wraps around some histoproteins. And it leads to a larger structure scale. And so it's basically showing how you can take DNA, which contains an incredibly large amount of information, and twist it together to form the DNA, and then wrap the DNA around things, and then finally, package it in such a tiny space. That's the other thing that truly amazes me. As far as density storage for information, humans have computers beat pretty handily. Not our brain, not memories and stuff, but the sheer amount of information coded in our cells and how many different and how we have a copy of it in every single cell. That's another one of those things that amazes me. It makes me just ponder how we can ever have any hope of understanding it all. Although, I mean, we're making humongous strides. It used to be that they didn't even know DNA was what controlled heredity and, passing on of traits. And now we know it. Not only do we know it, but we can actually measure it and find out all 500 million base pairs that make up any given individual or person. And so, that whole human genome project, to me, that's just amazing that you can do something like that. Okay, getting back on track. So we want to talk about what's next? DNA replication. So you probably have much more of this in Todd's class, I'm guessing, from already just hearing some of you mention or talk about things. What we really want to know or kind of focus in on, or what your book focuses on, is a couple definitions and then a very simple understanding of the process. And heredity, we've already mentioned, is just basically the process by which traits are passed along. Physical and mental. Passed from parents to offspring. Heck, that alone boggles my mind, how many different pairs that we can come up with to make people. I mean, if you look at, parents and their kids, you can see a lot of similarities, and you can start to see differences, and then just the combination between the two gives you almost an unlimited number of differences. It's kind of like looking at color in horses. I'm sure Jess can probably tell you a lot more about that than I can, but trying to follow the genetics of color in horses is ridiculous. It's a very complicated process, and stuff like that. We would call, what we call the genome is basically all of the heredity material. And so that would be the approximately 500 million base pairs. And then the smallest single unit that we talk about is what we actually call a gene. So that's the smallest segment of DNA. Oops. Oh good, it didn't crash. Small segment of DNA that controls the formation of RNA. And that is usually 3 base pairs. Again, normally you guys have this in high school. Your book talks about it, makes a small mention of it, and then kind of pushes it further out in the chapter about how that genetic code is done through or goes from DNA to RNA. So replication then itself is simply the process for duplicating DNA. And if you're following in your book, it's figures 31.10 and 31.11. They're kind of useful for understanding it. Essentially what happens is we have 2 strands of DNA, and they're split apart. And those 2 strands form a template strand and what we call the complementary strand. Complementary strand. So this is the original. This is the new strand that's being built based on that original template. And despite how simple pictorially the process is, meaning that if I take my strand of DNA and I split it apart and I split it apart and I'm making 2 new strands of DNA. I don't know, did Todd draw that picture? He probably draws a better one than me. So we have one of the strands being built from where the split is to kind of where the zipper connects. And then we have this other strand that's being built here. And the difference between the 2 strands that are built are this one is continuous, and this one is built in small segments. And then connected together. So this is called continuous synthesis, and this is called, I didn't put the word down, but I should probably put fragmented. And what you can, so as that DNA strand is being broken apart, you can really only have one of them being made continuously, and then that other second one is made in chunks that have to be stitched back together. And so when I say that it's simple pictorially, meaning you can easily imagine 2 strands being pulled apart and then the synthesis occurring on either strand, it is actually technically one of the most complicated enzyme-controlled processes in the body. Meaning that the enzymes that are responsible for breaking the strand, for copying the strand, and of course checking for errors, there's a huge number of them. Too many for us to ever want to bother studying in a 100-level class. I would argue that you could probably have an entire graduate level class on DNA replication where you literally talk about which particular enzyme controls what particular aspects, focus on the actual chemical reactions that are happening and things like that. So what I'm trying to get trying to get across to you guys is that conceptually, DNA replication is pretty easy, meaning the breaking apart of the strands. In an actual, truly understanding the process and knowing every nitty-gritty detail in it, very complicated. And I guess maybe we could point out that probably every topic in this book is really actually very complicated when you get down to the nitty-gritty details of how, all the chemical reactions that go into making it and stuff like that. That's the other thing that boggles my mind about biology is just the sheer amount of information you have to know just to understand DNA replication. Whereas you can understand huge chunks of chemistry, it seems to me, by knowing a few simple rules. And that's just because these processes are so complicated. Like, for instance, what?
So which side is the
In this case, this well, I kind of poorly said that. So it's not that this is not supposed to represent the split.
Okay.
Sorry. So this would be our template strands. And these would be the complementary strands being made together. So comp. I don't have enough space to fit the entire word in there. So since the book doesn't cover it in any detail, you have to know it at about this level of detail, that it simply splits apart, that one of the strands is continuous, one is complementary, that there's a very low error rate associated with it, 1 in a billion. Wouldn't you guys like to have an error rate of about 1 in a billion in life? I'm pretty sure I can't even type a complete sentence on the computer without having an error rate of 1 letter per word when I'm doing things. Okay, so we brought this up earlier, but I don't know where the best place to kind of put it in the notes is. This is kind of our transition from DNA to RNA. And what I want to focus on are, what are some things that are the same about them and what are some things that are different. So let's take a look at some differences first. What are the main differences between DNA and RNA? You guys have already mentioned some of them already. Number of strands. So DNA is 2 strands. RNA is 1 strand. And why, and since it's brought up, it's a good point to maybe ask that, why is 1 2 and 1 1? It kind of leads into their purpose too, so what the heck. So why is DNA 2 strands but RNA is 1 strand? Jess, are you going to survive? It seemed worse than yesterday. So the main thing is that DNA is used for information storage. And so the 2 strands being complementary to each other kind of make it so that that storage is very secure, meaning that one strand isn't changed while the other one is, or something like that. Whereas RNA is used for building proteins in the end. Building proteins. It's also used to transfer the knowledge. And so the complementary nature, the way that kind of works for RNA, is that since RNA is a single strand, it matches up with other small segments or small pieces of other molecules to act as the complementary part. And so they're kind of like the halfway part, meaning if they were complementary, they'd just match up with each other and nothing would happen. That's why DNA is great. It's paired up, nothing breaks it apart except for when they're transcribing it, and so it's pretty solid and stable. Whereas RNA, you can kind of think of as, it's got that one copy, and then lots of other molecules can interact with that. And because lots of other molecules can interact with it, because it's broken up into small parts and has many different roles, it's not a very secure form of storage information, since all of those molecules can be changed. At least to me, that's kind of why it is. It's mostly like a security issue. What are some other differences, too, that we've already talked about? I'm thinking, yeah, T and U. And why would it be important to have a different base on DNA versus RNA? Yeah, it's like a little flag that says I'm DNA and a little flag that says I'm RNA. You can't confuse the two of them, can you? Now there are other flags on them too that are differences, but that's kind of one of the big ones. What's another difference structurally wise? Big one, why is one D and R, one R? Yeah, so ribose, deoxyribose, which basically means that this is missing the 2' OH group. Because it's, I mean, if we really put the real name, it's actually 2' deoxyribose. So that's another little flag that you can kind of point out. I've got, do I have one more difference? I should have 5. That's usually how many I count to. Oh, this one's kind of tough. I'll give it to you guys. DNA, and this is my own notation, is just DNA. RNA is usually heavily modified. So that's, again, kind of going back to that 1 and 2 strand thing too. DNA is for information storage. You don't do anything to DNA other than copy it or transcribe it or replicate it. Instead, RNA is the thing that if you need to make a change to it or need to modify it or need to do something different with it, that you do it. So you never directly damage your original store of information. You only modify something afterwards. It's kind of like in chemistry class. You're never ever supposed to dump the bottle or the contents of something you've taken from the bottle back in the bottle because you may have contaminated it, right? And so what we generally do is I set out little bottles of stuff, and I have big bottles sitting in, you know, the back lab, and I never dump those little bottles back in the big bottle, because all it takes is one student to either accidentally put the wrong thing in there or maliciously put the wrong thing in there. Although I've not noticed too many malicious students here. For things to kind of go wrong. I would say most of them are things that get mistaken. You put the wrong pipette in the wrong place. And probably 9 times out of 10, if you swap pipettes, it won't make a big difference in your results, but that 9th out of 10, or that 10th time, it does. So those are some of the differences. What are some of the things that are the same? And I'm not so sure it matters in the grand scheme of things to focus on so much of the sameness. Although, you know, there are things that are kind of that's useful for it, I guess. So I would say the biggest thing that's the same about them is that DP, 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 and in RNA it really is RP, RP, RP. But what I'm trying to get at is that the backbone, at least for all intents and purposes, is the same enough that when you're doing transcription that you can match the pairs up. So the missing OH group between the difference between the D and the R's in that chain are very small. The other important thing that's kind of the same about them is the hydrogen bonding between base pairs. I don't know why I'm making 2 lists, because I'm almost really, you know, like this one should be, if these are things the same, they belong to both of them. So it's the hydrogen bonds between both base pairs, meaning that they're both capable of forming those hydrogen bonds. That means that when you match up DNA and RNA, that you can very easily keep them that way. So when you're doing transcription, when you're going from the DNA to the RNA, they pair up really, really well. And so there's enough things that are the same about them that they match up pretty well for transferring the information, but there's enough things that are different about them that the body can kind of have enzymes that recognize which one is which, and so use them for their different purposes. You never see, for instance, RNA being turned into DNA or RNA being used to store information. Although, you know, a certain part of me says it wouldn't be surprised if that actually happened. Some research right now is showing that RNA can actually function as enzymes in the body, and that they think that potentially the first enzymes were not really proteins, but it was basically all an RNA world at that point. And RNA performed almost all of the functions of the body until better things were evolved or developed or given or whatever. I mean, I'm not going to get into that big debate. So there are some differences between DNA and RNA. There are some similarities between DNA and RNA. So how do we go from DNA to RNA? Does anyone know what we name that process? I've actually said the word enough times. Yeah. Transcription is the process by which DNA is turned into RNA. And we're not going to go into the details of that process. It's one of those things that's more complicated than what we really care about. Instead, what we want to focus on is the types and roles of RNA. And even here, we're going to shortchange one of the three types of RNA, and really maybe even consider it two of the three. Meaning that this book and the level it's written at and the level the course is at is asking for the broad appreciation of the subject with some depth in details here and there, but certainly not all of the depth that could happen or one could talk about. So what are the three types of RNA? Most people know that. mRNA, tRNA, and rRNA. I guess maybe, I don't know what order it matters if we talk about them. mRNA is called messenger. And this is usually, for whatever reason, those things stick in people's head because of the M, R, and T pretty much tell you what it is. And what this really does is this transports the DNA information. And what I mean by when I say we're going to kind of shortchange it is if we kind of look at it, it's approximately got a very simple structure. And it really is just complementary to the DNA. Meaning nothing happens to it other than it's, I guess, really is broken into smaller chunks. So it's kind of like if you have a book that's got 1,000,000 recipes in it and you pull out one recipe to use. It's still the same recipe as in the book, hasn't been modified, but now you've only got a chunk that describes just one recipe so that you don't have to try to deal with the other 1,000,000 pages in your book. So that's one type. What's the second type? Maybe, but I'm going to talk about rRNA next, which I don't even know why I asked you that because I was going to do it in this order anyway, meaning that we're not going to say much about rRNA, but we are going to say a lot about tRNA, so it makes it more sense to do it in that order. What does that stand for? So what does that mean? So essentially what they are is they're the machinery to synthesize proteins. And we'll look at that process in some detail, not today, but Monday. And what they really are is they're about 30 to 35% rRNA, and they're about 60 to 65% protein. So I kind of like to think about them as the glue that holds the machine together, maybe. So what rRNA does is it binds through hydrogen bonding and all those other interactions that we've talked about, intermolecular forces included, but mostly salt bridges and things like that, with the proteins, and it holds it all together in the exact specific shape that that protein needs to do to function in making or synthesizing proteins. And so it's kind of like taking the instructions to do the recipe and chopping them up and giving them to a bunch of different people or distributing them in such a way that then we've got an assembling line for making the proteins. And I should point out that We said that this has a simple structure. This is highly complex structure. Your book skips over it, and I remember last year when I was a little bored one day, since I'm weird, I do things like this. I was just curious about why your book always skips it, so I Googled ribosomal RNA and stuff like that and tried to see if there's any small piece we could add to class. And really it just starts off with, here's 5 bajillion reactions and interactions that occur. And it's like, I know why they skip it. It's just not anything that anyone who isn't getting a PhD in transcription really wants to know. Now, the third one, they actually give you a bunch of detail on. So this will take up a good chunk. So if you're at the tiny bottom part of your page, you may want to stop and start a new page. This, of course, stands for transfer. And what does it do? What does transfer mean? Not quite. It actually brings amino acids to the ribosomes to synthesize proteins. So what the ribosomes do is they're the machinery, right? The mRNA is the template, so that would be going through the machinery, and then what the tRNA is doing is it's bringing the amino acid and matching it up with the appropriate part of the mRNA ribosomal machinery, and then the ribosome machinery kind of stitches it all together. So I think of it as a giant assembly line. And so, you've got all these machines, and have you ever seen like the assembly line for a car plant? It's got little robot arms that are swinging around, and it goes and drills a hole in something, and then it puts another part and smashes them together and things like that. That's kind of what I think about as the ribosomal stuff. The mRNA would be the big blueprint that says what order everything goes, and then you might think about the tRNA. That's like another piece of machinery that either brings a part or it's the human that carries something over and dumps a bunch of stuff in the bin. I don't know if that's a good example or not. We're going to kind of cover this in intermediate detail. Meaning that, I guess, to me, this is also one of those things where it's like, if this is the only thing that they give detail on, guess what's probably going to be on the test? Something about tRNA. There might be a random question that says, what is RNA composed of? What does RNA do? What does mRNA do and stuff? But since there's very little detail on those, it would be a quick, short, simple question. Whereas something on tRNA, because there's a lot of detail, means that there's things. Or more things. And the reason we want to do this is because we can see how the structure of tRNA leads to its functionality. Now, I'm going to refer you, and I'll maybe sketch a picture of it, but certainly page 868, figure 31.12 is probably something that you would want to look at. And I think I have it. Otherwise, maybe I forgot to put it in. Let's see. I hope I put it in. I have this feeling that I didn't, though. The one on page 868. Well, that's a bummer, because that means, why would I have not put it in? Unless I had this lecture split into 2 parts, in which case this would be in the second part, and that's why I didn't put it in. What I've been noticing is that really every year I cover a different amount of material in a different lecture. Like you notice that, for instance, on the homework, the last question, well, it wasn't the last question, but one of the questions was draw the DNA strand, right? Or a part of the DNA strand. Well, this year I didn't cover it. Last year, apparently, I did get that far. So there's always little mismatches like that. I think they will always remain. So let me sketch the picture then. What you get is something that looks kind of like a 4-leaf clover. And this is supposed to match up and be a lot more symmetrical than I've kind of got it. And so there's kind of 4 parts to it. And so this represents our amino acid. This is supposed to be our tRNA strand. And so what's happening, or what you can think about it is, here's our little different bases. And so first thing that you should realize is that the specific shape is due to the hydrogen bonds between the base pairs. So, you know, on one side we might have an A, well, here, I'll do this, AU, CG, that's what's holding those parts together. And so, for instance, you know, there's no hydrogen bonds holding that center part together, and there's less hydrogen bonds holding sort of the leaf part of it together too. And each of the arms kind of has a different function. So this arm, let me put it in red, so because I'm trying to cram it all on one page, which is stupid. This arm basically is called the terminal arm, and basically it binds to an amino acid. And there is basically one tRNA for every amino acid, at least. Some of the amino acids actually have more than one tRNA associated with it, but it's basically mostly a one-to-one correlation. So this is also, oh, I see. This side is called the anticodon triplet. And so there's really 3 base pairs on that side. And this is complementary to the mRNA. So one side of the molecule binds to an amino acid. The other side of the molecule binds to the mRNA. So that's how we get the amino acid to the appropriate spot, kind of on the mRNA, or how we can put the appropriate amino acid order together by following the appropriate code. And see, this is one of those things where your book is, I think, out of order, because your book doesn't talk about the DNA or the genetic code until the next couple sections. And so maybe I'm skipping ahead, but I guess I always assume people know this. So you guys know that 3 DNA base pairs or RNA base pairs code for a very specific amino acid, right? You guys are familiar with that? And so what we're saying is that this is codes for the amino acid, and then this is what binds to the amino acid. And so it's acting as the intermediary between the mRNA and the amino acid. And then these other 2 arms are basically hooks for the ribosome and other enzymes. So one part of the molecule binds to the mRNA, one part of the molecule binds to the amino acid, and then the other 2 arms are what are attached to or that the ribosomes can attach to, so that they know how to get into the proper orientation to catalyze the binding of the 2 amino acids together. And so if you look at it, this is a theoretical model. If you go and look on the page, and I'm not even going to try to draw it, you'll find that the actual 3-dimensional representation is much different, and the 2 loops that are shown as being kind of the control arms, I guess you could say that, they're the control arms, I don't know. I make up my own vocabulary for some of it, are sort of in one part of the molecule. And so really you've got 3 things going on when we talk about on Monday how to make a protein or an amino or an enzyme, or how to stitch together. You've got the plan, that's this part down here, that anticodon triplet. You've got the part, that's the amino acid that it's carrying, and then you've got those control arms, that's where the ribosome hooks on so that it can start stitching all those amino acids together. And I don't know, to me, this is just one of those kind of amazing things, how all those hydrogen bonds and everything else has to line up perfectly, and then all the other parts have to match up with, I think the anticodon triplet's probably pretty easy to match up, but like the part that binds to a specific amino acid has to, you know, have a very specific shape so that that amino acid gets locked in place, kind of like the active site. And then the arms that the ribosomes and stuff interact with, those also have to have a very specific shape. So everything has to be picture perfect. And to me, the sheer beauty of how it's all done like that is amazing. I mean, it's just like, how does it even get that way? So those are the three parts of the RNA that we want to talk about, or the three types of RNA. And then we had said that, and I'm going to go back to blue. I always feel like I'm shouting if I write in red. That process of transcription, again, is going from DNA to RNA. And the process is complementary in nature. So one of the things you'll notice is that this idea of complementary, depends or occurs over and over and over again. The two strands of DNA are complementary. The DNA and RNA are complementary. The ribosome and the messenger RNA, I'm sorry, not the ribosome, the tRNA and the mRNA have to have complementary portions. That basically how these things lock together are continued. I kind of sometimes like to think about it how Legos are complementary, right? Meaning you can take two Legos and stick them together, and you can stick any two Legos together kind of because they have little dots on the top and little holes in the bottom. It's under strict control, meaning it's regulated by enzymes. And it only occurs small pieces at a time. That's that idea of that you might have 1,000,000 recipes in your book, but we only have one recipe at a time that we go and cook up or that we look through. And there's a considerable amount of modification that occurs. Considerable. I just feel like I shouldn't there's no D in considerable. I wonder why it looks considerable amount of modification. So one of the things that we said is a difference between DNA and RNA is DNA is pure. It pretty much is exactly the structure we talked about. But by the time we get to that RNA and the very different types, most of them have significant changes made to them. Specifically, the rRNA, and the tRNA have considerable changes. They tend to start out as large, and then they're modified, and then they're kind of trimmed down to size. And there's an entire class, like I said, of enzymes, meaning it's strictly regulated, that do that. And so some of the types of modifications that occur is usually the bases is what's modified. And sort of the three most common ones are methylation. Methylation. Can you guess what that is? Just from the name, methyl. Right, so so it means one, but it really means CH3, right? So CH3 is added to them. There's saturation. So that's where you take a carbon-carbon double bond, and of course, what do you do to it? Add hydrogen to break it into single bonds. So that's kind of like how saturation works in lipids. I mean, it's not the same process, obviously, but it's the same word, meaning saturated means that we take double bonds, which it would be unsaturated, and add hydrogens to them to make them saturated. And then there's the isomerization of ribose, in which basically an isomerase is used to convert ribose into a slightly modified molecule. So what this does is this takes the DNA, which is just the blueprint, and starts adding a bunch of things to it that have functionality, that change the shape of the molecules, that change sort of the information that they contain. It no longer becomes a blueprint, but it can become either a piece of the machinery, meaning the rRNA, or it can become that tRNA, which has got all the different parts that kind of connect all the machinery and the part and the amino acids and the mRNA all together, or at least it interacts with all three.
So is that the post-transcription modification?
Yes, post-transcription modification. So we could call this post-transcription, which is a good thing to bring up, Brittany. It's one of those things where, you know, it kind of slips my mind the way I've got it written down or thought about. The idea is that the modification happens after the transcription occurs. One last topic I want to cover, just to get it out of the way so that we can pick up at sort of a topic. And I'm not going to go into a great deal on this topic. I want you guys to just realize some complexity. There's an entire series of things called small RNAs. So when we tell you there's 3 types of RNA, we're lying to you. Or at least we're simplifying things down to you. I want you guys to get across the idea that it's far more complex, meaning there's more than 3 basic types of RNA. And this is one of those things where, you know, 20 years ago or even 10 years ago, I don't think they would have had in the textbook. And I also don't think they would, you know, we didn't have a very real understanding of it. A lot of people just thought that all of these smaller or extra RNA fragments that came, you know, that they would find in cells or would crop up in their experiments were simply drunk or detritus or the breakdown products of things. But really, they actually have a lot of different functions. They control the genetic information. So they actually act as both enzymes, and they act as inhibitors, and they act as activators. So one of the reasons people think that our RNA might have been the first molecules of life sort of built is because they can serve all of those roles, whereas in our current setup where, you know, most things are enzymes, you don't use an enzyme to act as a catalyst or an activator very often. And so we can have a bunch of different categories. We have miRNA, which stands for microRNA, and that's about 20 to 25 nucleotides. So remember, when we're talking about genetic information, we're usually talking about thousands, if not tens or hundreds of thousands of base pairs. So these really are very tiny. That's why they're called micro. And they're found in the cytoplasm. And basically, they stop translation. By blocking ribosomes. So in some manner, they interact with the ribosome to tell it when to start and when to stop something. We have small nuclear RNA, so snRNA, small nuclear RNA. This is about 60 to 200 nucleotides. So again, very small. And these are what control post-transcription modification. Post-transcription modification. So they're what specifically would add a methyl group, or they're what specifically would tell the molecule to saturate a double bond, or they would specifically, you know, direct the isomerization of the ribose. We can have small nuclear RNA. Oh, I put the abbreviations first. Small nuc. Now I can't read my handwriting. rRNA. I think it's like that. So oh, I see. Small nucleolar RNA. These are about 70 to 100, and they control or process ribosomal RNA. So they specifically kind of direct the process by which ribosomal RNA is built from the parts, from the rRNA and the other protein parts that go in with it. And then the last one is called small, I guess I'll abbreviate it first, siRNA. I love the names they come up. Small interfering RNA. And these are only about 20 to 25 again. And they stop transcription by triggering mRNA destruction. So notice that they can act like enzymes, meaning I don't know if they necessarily speed up the reaction per se, but they certainly control when the reaction occurs and how often it occurs. They can inhibit things, and they can actually act as activators, meaning they can actually build things or cause things to occur. This is a hot field. Yep. This is a very hot field of research currently, meaning if you look in your textbook, they have the most rudimentary discussions of it. If you start reading any literature out there, and I don't know if you'll run across this when you're looking at your dental hygiene stuff or not. I don't know the detail of level. I do know they make you write some, I won't say research papers because you're not really doing the research, but you're sort of reporting on other people's research, or at least trying to think about it critically. And one of the topics that you might come across is something about this. So this is a hot topic, meaning there's lots of money in it. It's new, it's exciting. People see lots of potential for it. And the idea is that you can use this small RNA to combat or to affect metabolism or metabolic pathways. And I kind of screwed that up. So maybe we should say just affect metabolic pathways and combat diseases and things like that. For instance, you can use the siRNA to block protein production. So, for instance, you know, where we currently use a drug that might, say, block the COX-2 in when you're turning arachidonic acid into things like that, into its thromboxanes and things like that, in that pathway we talked about. You know, currently we would use things that are protein activators or inhibitors to control things like that. Well, you might be able to use small interfering RNA to do that. So this might be another approach that we could block protein production. You can use it to match up to specific RNA, and that actually leads to a double strand, but then your body immediately says destroy it. So your body understands and knows what double-stranded RNA is, and if it encounters it, it destroys it. So for instance, if you had a pathway in the body that you wanted to affect, potentially if you put in the complementary strand of RNA, you could destroy that specific protein being built or made. So that's another way that you could do it. You could stop the production of it. The list of diseases that I found when I looked at it is pretty impressive. It's things like using cancer to kill solid tumors, to block creation of lipoproteins so that cell membranes can't be made, viral diseases, macular degeneration, hepatitis, atherosclerosis, Huntington's disease. I don't even know what that one is, to be honest. I just was on the list of stuff. Does anyone know what Huntington?
I don't know what it is. I couldn't tell you though, because we talked about it a little bit in biology, and I know someone whose friend, like everyone in your family has it.
It's genetic, right?
Yeah.
Isn't that the one that kills you, like, when you go late 30s?
Yeah, I can't remember
Yeah, I know it's not Huntington's disease, but that just reminds me of a family I heard about where, yeah, basically the guy married the lady, and all of her kids got the disease. So he's watched his entire family die basically from this disease, and it's just like, wow, talk about kind of a shitty life. I mean, don't get me wrong, life's still worth living, but it can't be easy watching your entire family die before your eyes. So anyway, that's it for today. That's a good spot to break. We will come back and we'll talk a little bit about the genetic code. Honestly, you guys already know more about it than we need to know probably. We will talk about biosynthesis of proteins, and then whatever leftover time, I usually just randomly talk about some topic that sounds entertaining or that I've read a news article about recently. So we should finish up with this chapter on Monday. So let's make homework 31A due Tuesday for sure, and 31B, I think we covered enough to make it due on Tuesday also. And then for lab next week, if you want to start ahead of time, at least thinking about it, we're going to cover chapter 32 on nutrition, except I'm a lazy SOB, and because most of you have had Sarah's nutrition class, you know more about nutrition than I probably want to know. So you guys are going to teach me nutrition. So what we're going to do on Tuesday and Thursdays, I'm going to give you work days, essentially, where you can prepare a lecture, a homework assignment, and a list of test questions that I will actually use on the test. I'll put some of my own questions in there, but I guarantee I'll use at least one question from each group, so you'll know, 4 or 5 of the test questions ahead of time, right? And you will be able to look at that. I'll also hand back tests on Monday, since I've got one slacker here that still has to take it this afternoon or later this morning or whatever time you end up picking.